the main interest of our laboratory has been studying the impact of glial cells into neurodegeneration. And I would like to provide you with some information today, which is mostly unpublished, showing that glial cells can be primarily damaged in the neurodegenerative diseases. Specifically, I don't think it works. I am trying both ways without success.
Here for next.
Okay.
Okay. Back.
Thank you. So specifically, I would like to show you three main ideas. The first one deals with the signaling of beta amyloid directly on astrocytes. The second about the effects of beta amyloid in oligodendroglia and myelin. And finally, I would like to show you the role of astrocytes in Parkinson's disease spreading. So there is an overall consensus that one of the major features of Alzheimer's disease is the aberrant processing of the beta, of the amyloid precursor protein. So in this, I cannot see the pointer, right? Well, anyhow. So oops. Sorry. So environmental factors, so genetic factors, result in the overproduction of amyloid species, and this, when released, can cause the accumulation in the extracellular space, which ultimately lead to the formation of plaques of different types. So this A-beta oligomers can be synthesized in the laboratory and assayed in different cell types. So we use this preparation of synthetic oligomers, which in a Western blot show that they are formed mainly by tetramers and pentamers. And when we look at the electron microscope, we saw that the majority of them have this particular rounded form, suggesting that they are mainly oligomers. And there are also a minority of fibrils, but the majority, which is the idea I want to emphasize when talking about this preparation, is composed by oligomers. So when we challenge initially neurons with these oligomers and record from them, we found that a beta amyloid would trigger currents which had the pharmacology of the NMDA receptors, as displayed here. And these currents would cause ultimately excitotoxicity. And we could evaluate the toxicity in areas which are particularly vulnerable to Alzheimer's disease, as the hippocampus and the entorhinal cortex. And what you see here in color is a marker which is propylium iodide, which intercalates into the DNA and shows which cells have the membrane damage and they are dying. And again, when you do the pharmacology of this cell death process, you see that it is mostly mediated by NMDA receptors, and since it is blocked by the specific antagonist to those receptors, and also by AMPA receptors. We wonder, after doing these initial experiments a number of years ago, what would happen in astrocytes. This worrisome about astrocytes is not new. Since more than 100 years ago, Nicolas Achoucarow, who he was a pioneer in the study of glia at the turn of the last century, working with Alzheimer's. Himself found that astrocytes form particular processes which surround the neurons, and this occurs again in vulnerable areas of Alzheimer's disease like the hippocampus. We very recently observed that these formations, when you use higher resolution microscopy, they are indeed present in transgenic mice, which I will talk later about. And when you go into the electron microscope, you see that there is a net surrounding the cell bodies, which seems to separate the synapses from that cell body physically by astrocyte processes. So we did studies of signaling with the oligomers I showed you before in different preparations in acute hippocampal slices, firstly isolated astrocytes, and also in primary cultures. In all of them, we saw that the oligomers would generate an increase in the intercellular concentration of calcium. By the way, the shape of the signal differed depending on the population of cells we were using, on the origin or the preparation we were using, but in all of them, such a signal was present. Elena Alberti, making a tour de force, pharmacological tour de force, trying to characterize those signals, found that the signal was not mediated by glutamate receptors nor by purinergic receptors, and that it was blocked by using blockers or antagonists of the inositol triphosphate receptors in the ER or the ryanodine receptor. In addition, and interestingly to us, she found that blocking integrin beta-1 would reduce greatly the signal. So, to make a story short, what activation of beta-1 integrin by oligomers does is activating the NOX2 oxidase, and this activation leads to an increase in the expression of the NOX2 itself, and in addition to the generation of reactive oxygen species, all of which are blocked by using a non-selective antagonist of this family of NOXes, or more specific antagonists of the so-called NOX2 oxidase. And this was done using a lot of beta amyloid, 5 micromolar, and you may question this and say, well, is this physiological? So to go a step further, these experiments, we produce native oligomers by transfecting N2A cells with the wild type APP or the APP with the Swedish mutation. And using this supernatant conditioned by these cells, and at picomolar concentrations, we found again that oxidative stress was generated in astrocytes. So I don't want to give you all the details about this signaling cascade. There is a poster dealing with it, and just to sum up, what A-beta oligomers do by interacting with integrin beta-1 is to activate the PI3 kinase, and all the cascade initiated at this level leads to the increase in expression of the NOX2 oxidase and the generation of reactive species, the increase also of the integrin beta-1 in the membrane, and ultimately to astrogliosis, and also to astrocyte cell death. So what about integrin beta-1 and astrocytes? Integrin beta-1 is expressed quite abundantly during development, but in the adult brain there is not much. Indeed, it is very difficult to locate it. You have to try many different antibodies. And what we found using one of those antibodies, that there was diffuse but prominent staining of the CA3 area and other hippocampal areas. And when we zoom in by doing electron microscopy, we found that indeed the integrin beta-1 was expressed in perisynaptic astrocyte processes. So it is possible that this integrin is playing a role, an important role, in the tripartite synapses, which is just there. In addition to perisynaptic astrocytes, we also saw expression in postsynaptic spines. So all those experiments, the ones I showed you briefly before, were done in vitro. So how about in vivo models? We used two options. The first one, and the most obvious, was to inject the oligomers into the hippocampus of a normal rat. And we found, again, that there was an increase in the GFAP, there was astrogliosis, and there was, again, an increase in the NOX2 levels. And when going to a more reliable model, the so-called triple transgenic mice, which recapitulates many of the major features of the disease, including the presence of plaques and defects in synaptic communication, we found that indeed in the hippocampus and other areas there was an increased expression in the levels of integrin beta-1, NOX2, and GFAP, the critical three molecular components of the pathway I am pointing out today to you. So, and this increase was more prominent in the older mice, so basically around 18 months. And when you compare the increase of those molecules with the levels of oligomers present in the sample, there was a clear correlation in all instances. So how about the histology? In histology, again, we're showing that there is an increased expression of all these markers around different types of plaques, like neuritic plaques here, or core plaques. So, and how about the EM? So we found that indeed astrocytic processes were abundantly marked with antibodies to the peptides, to the amyloid and this occurred around neuritic plaques and also around soma, which were undergoing degeneration. And when we look at the synapses, or around the synapses, we found that this peptide was also labeled, was present within these perisynaptic astrocytes. So it may well happen that this signaling and this accumulation of beta amyloid is impairing the tripartite synapse. But this is an idea, a working idea we have to work out, and we are planning to examine it by doing electrophysiology in the near future. So how about the post-mortem Alzheimer brain? So again, the same thing. We find that integrin beta-1, NOX2, and GFAP is increased in average, at least. There is a subject heterogeneity, but in average there is an increase of the expression. And when we do the histology, again, astrocytes around the different plaques show an increase in the three markers, GFAP, NOX2, and integrin beta-1. So, and just to end up this block of experiments, we did a preliminary evaluation about the different stages of the Braak stages of Alzheimer's disease, and found that there is like a trend of an increase in as the disease is more severe in the expression of these markers. So to leave you just with a simple view of all these studies, it seems like as Alzheimer's disease progresses, there is a astrogliotic phenotype, which again advances, and this is driven, at least in part, by the increased abundance of amyloid peptides. I would like to briefly go to the other parts of my presentation, and the second one is the one dealing with signaling on oligodendrocytes. So this is a very simple scheme as to how an oligodendrocyte progenitor matures. You see, without going into the markers, we characterize the process that initially they have a very simple arborization and then a very, very elaborate one in which there is plenty of myelin and myelin proteins. And this is how it looks when you stain for them with those markers. So what we found is that submicromolar concentrations of oligomers increase the amount of myelin components, and that this increase could be attenuated or blocked by using blockers of the SRC kinases. And that experiment was done in dissociated cultures, but when you do those experiments in organotypic cultures, for instance, of cerebellum, you find exactly the same result. So it's not, this is a more reliable preparation than working in dissociated cultures, and it seems the same happens. So again, we have worked out the signaling of the oligomers onto the membrane of the oligodendrocyte. We don't know which is the receptor, in this case, to the oligomers, but we do know, and the details are provided in a poster, which we have here, is that there is an activation of the SRC kinase, and which in the PKA kinase, which drives the activation of CREB and ultimately leads to the increase of the basic myelin basic protein, which is a major component of the myelin. Interesting, we also found that the oligomers play a role in repair of myelin lesions. And without going into the details of the data, the experimental setup illustrates here that with the toxin, which is called isolecithin, you can demyelinate the preparation I showed you before, and then if you incubate the slice with beta amyloid, you get a beta recovery of the myelin that if you do not. When again go to the myelin tracts of the triple transgenic mice, we see that regardless of the area we look in, corpus callosum or hippocampus, we have an increase in the levels of myelin. And when we do the histology, for instance, of the hippocampus, we see such an increase, which varies depending on the area we are in, and is particularly prominent in CA3 for whatever the reason. If we do the electron microscopy, we see alterations in the myelin in the triple transgenic. For instance, we see thicker myelin sheaths. There is hypermyelination. Indeed, we see changes in the appearance of oligodendrocytes, and we also see signs of de novo myelination. In there, we wonder what was the impact of these changes in the function. And I want to show you very, very recent data obtained last week, indeed, using a test, so in which we put the optic nerve of the transgenic mice or the control into a chamber. We stimulate it from one end and measure the so-called compound action potentials at the other end. And what we get is this kind of recordings, and we can evaluate the first peak when it is coming, the latency, and also the number, or an estimate of the number of actions which are activated. And what we see is that there is a reduced and impaired impulse conduction in the triple transgenic, which is quite marked. And the only question is This is very preliminary. The number of experiments is still very small. It's 4 in each of these. So what happens in the human brain is the same thing again. There is signs, clear signs by Western blotting that there is an increase in the myelin content, as you see here, in the brain as compared to the controls. And when you do a preliminary study again of the different groups according to the BRCA stages, it looks like there is again a tendency to have a higher hypermyelinization as the disease advances. So, and just very briefly, the last part of my talk deals with another astrocytes, another property of astrocytes. In collaboration with the laboratory of Urban Bessar in Bordeaux, we have the chance to get Lewy body extracts which were taken from patients with Parkinson and challenged them in neuronal cultures and astrocyte cultures. What we found is that after incubating for a few days, we could detect human synuclein within the neurons and also within the astrocytes. So, interestingly, neurons would suffer. I mean, the extract would be toxic to neurons, as you see here, but not to astrocytes. This is a very interesting feature. So, astrocytes are resistant, but neurons are vulnerable, as expected. So, the way the synuclein reaches the cell, the inner part of the cell, is through the endocytotic pathway, as you can see here, that they are with markers of endosomes or lysosomes, which are introduced. They are using baculoviruses. And this is also the same process in neurons and in astrocytes. So we wonder whether the synuclein containing those extracts would jump from one cell to another. And for that, Fabio Cavaliere, who we saw in a poster with the details of this study, exploited these microfluidic chambers in which you can put one cell type in one side and another in the other, and then through channels, there is growth of processes, and you can load in one side the extract, the Lewy extract, and then put pressure at the other side so that if there is transmission from one side to the other, it would be through an active process. And what he found is that there is transport. If you put neurons here, from neuron to neuron, there is transfer from astro to astro, and there is also transport from neuron to astro and the other way around, as shown in here. So this means somehow that astrocytes can propagate indeed or suggest the pathology. So finally, when you incubate with the extracts, the different cell types, astrocytes and neurons, there is an increase in the expression of the endogenous alpha-synuclein, which is quite shocking and marked. Yes, this is the message I want to leave you with. Beta amyloid alters astrocyte function via intergene beta-1, and it targets oligodendroglia and causes hypermyelination, and astrocytes are resistant to pathogenic alpha-synuclein but facilitate disease spreading. And this opens plenty of questions. Let me just point out only a few of them. So the first one is, can markers of glial dysfunction, and in particular the ones I was mentioning, GFAP, NOX2, or intergene beta-1, can be used as surrogate markers of disease by exploiting, for instance, the availability of cerebral spinal fluid. We don't know if something will lead to test, and we are undergoing conversations as to how we can do this effectively in a reasonable number of samples as a pilot project. So the second question is does beta amyloid pathology alters specific functions of oligodendrocytes, like the behavior of the tripartite synapse, or just the neurovascular coupling? We don't know that, but we are designing experiments to test specifically those points. And what is the impact of hypermyelinization in Alzheimer's disease? Does it affect the overall information processes? Does it contribute to the symptoms overall? We don't know. We have to do more experiments in the models we have. And finally, is the astrocyte syncytium a target for drugs attenuating the spreading of Parkinson's disease pathology? We don't know, but this is something we want to test, and we are on the way to test. So finally, to acknowledge the work of the collaborators, especially Elena Alberdi, who has led the work on beta amyloid signaling, and Fabio Cavalieri, who has done the experiments with the Lewy extracts. And also to our collaborators in our surrounding in the Chúcar Center, José Luis Zugaza, Estíbal Capetillo Zárate, Erwan Besar, and Pepe Obeso, a member of CIBERNED. And I thank you for your attention.